Everybody grab pizza.
All right, okay, cool.
All right, cool. Hi guys, we're going to go ahead and get started. I'm Weston. I'll be doing the newsreel for the day. So what we're going to do is we'll do newsreel, and everybody can grab their pizza real quick, and we'll have Neha come and talk. So we're really excited for that. So yeah. So thanks for coming, everybody. Just a quick newsreel on things that have been going on in synbio recently. So we'd like to do a little bit about industry as well as academics, and in industry, a few interesting things come on. So gene editing, sort of beyond the CRISPR battle, this Selectus basic company has filed for patent successfully, which has been maintained by U.S. Patent Office, that basically encompasses a really broad use of gene editing technology. So to me, this kind of just communicates that the CRISPR battle isn't the only battle going on in gene editing right now. So you can go ahead and read into that if you want to figure out exactly what they hold and what they don't. Odds are most of it's going to get shot down in the CRISPR area, but we'll see how that goes. But if you're sick and tired of competing with CRISPR, you can also just invent your own new gene editing technology, as Horizon has done, which actually has some really cool applications, and so it's transposon based. So it'll be interesting to see if that can in fact become superior, or at least a replacement, because the CRISPR space is so fraught.
How do we direct where it goes?
I don't actually know. You should check it out. Cool. So also in synbio industry, this Synthego has basically published this tool online. So if any of you guys do CRISPR, particularly in non-model organisms, they have over 100,000 genomes in this database now, and it's totally free to use online. So you guys should check that out. I think that it's costs money for companies, which is how they make money, but you should check that out. Oxford Nanopore, if you ever want to talk about that, talk to Isaac. He's like #1 super fan. Basically, Oxford Nanopore is this technology where you're able to use a protein to actually sequence DNA through a membrane.
A nanopore, you might say.
What's that?
A nanopore.
A nanopore, one could say. So there's a paper out in bioRxiv that basically shows these like ultra long reads, which is really exciting. And they also have this branch called Play DNA, where they're basically moving this technology into schools and allowing students with these little like USB ports. You guys have made it's a mini ion. Is that or minion? Or minion? Yeah. So basically, it allows them to play with those and sequence DNA, and then like actually use computers to analyze that, which is really fun. Close to my heart, The Economist has published an article on cell-free technologies that's really cool, particularly highlighting Vitro Synbio and Sutra Biopharma. It's nice to see this basically get out. They have a fun illustration here, so you guys can check that out. In the other things going on, gut microbiomes continue to be on the rise. So this Biomics has raised 24 million, and Synlogic, which has been a player for a while now, has recently merged and appears to be, they're in clinical trials as well. So check out that. So Ginkgo Bioworks also has finally showed some, so often people don't really know what Ginkgo Bioworks is doing with all their money, but they are making a commercial scale fermentation of flavor and fragrances, and they have in fact done that, and I think it's 50,000 liters, so that's pretty exciting. They're cooperating with Robert Ed to do that. And something that I didn't totally know about is this Rebel Bio, which is a UK like startup accelerator.
I think it's in Ireland.
And it's Ireland, okay, yeah. So it's in Ireland, and they have basically 5 startups that they highlighted in a really cool article, which we'll send out on the listserv. I noticed that they have a nice cell-free kit that's one of the 5, and then they're basically moving to a new generation now. So they're the oldest biotech or synbio like accelerator, and have some pretty cool programs going. So that's cool. In the research area, there was a paper that came out of synthetic genomics, Venture and Gibson, basically on this digital to biological converter. It's Nature Biotechnology, kind of a bit of a tour de force with their technology. It doesn't give a lot of descriptions because it's industry, but basically they go from synthesizing nucleotides all the way to like complex proteins using an in vitro transcription translation system, as well as sort of this whole suite of working with nucleic acids that you need to actually make that occur. So that's pretty cool. So you can imagine something more on demand. So this is with no human intervention whatsoever. So just like sequence in, protein out, which is pretty cool. So there's also some work on these synthetic mRNA devices, which basically are able to detect DNA or detect proteins in mammalian cells, like in vivo, which is pretty cool. So I haven't read a lot about this technology, but I think it would definitely be worth checking out. That's in nucleic acid research. Other things going on, tools for putting synbio in the body. A couple papers came out in there. This is still in bioRxiv, but it's out of the Domino lab. And basically they developed a thermostable Cas9 that's able to last longer in blood plasma, which is cool, so you can imagine that being more useful. And basically you can see this Cas9, its melting temperature is like quite a bit higher. So they mutated a few residues on it to make it even more thermostable and basically show that it can last longer in human serum. So that's kind of fun. And then in Nature Biomedical Engineering, we saw a paper on these self-adjusting gene circuits for insulin resistance, which is kind of cool. So they developed these cellular like nodules kind of things that you can actually implant into the body, and then they'll sense and respond to insulin levels. So sort of like a self-regulating, but it's made of cells, right? So self-regulating insulin pump, I guess, made of organic matter, which is pretty cool. So they're not releasing insulin per se; they're releasing hormones which can cause a cascade, which can induce insulin production. So it's cool. Always fun to see genetic recoding in something besides E. coli. So that string of papers came out in that today, or this month, and so this one's out of the Silver Lab. They basically use this like new DNA strategy where they actually use rolling circle amplification to sort of step-by-step drop in pieces of DNA to the genome to recode the genome. So they, I don't know, substitute 1,500 leucine codons or something like that, which is actually the most codons ever substituted in an organism. They didn't recode the whole Salmonella genome. This is in Salmonella. And so they didn't do the whole thing, but they did recode a pretty major section, and so they showed that you could do this more. Odds are they'll do the whole thing and they get another paper, so that's fun. And then the Church Lab came out with a quick thing in bioRxiv, basically showing that recombinating in Vibrio is useful. People are really interested in Vibrio because it doubles crazy fast, like twice as fast as E. coli, so basically that's really helpful for rapid prototyping. And then the Voight Lab with the classic tour de force. I hate these figures, but that's fine. Basically, you can't tell what's going on, but they're refactoring the type 3 protein secretion system in Salmonella and showing that they can do that, which is fun. That's like this way. Civic engagement opportunity, we just want to make you guys aware of. Congressman Jane, how do you say her name? Schakowsky. Okay, cool. She is our congresswoman, and she will be visiting TJ's Commons tomorrow. So you guys should check that out. In particular, we wanted to point out like this might be a good opportunity to discuss what's on our minds as far as like science policy and what we think is important, and sort of like getting the truth about what we do out to politicians, which is increasingly important in this climate. So yeah, that's all I have. Thanks, guys.
All right, pizza break, I think.
Do we have pizza?
I will confirm.
Is there pizza for the pizza break?
Pizza break.
All right. It's not looking good.
We may have
Break right now.
end of meeting pizza.
Might be an end of meeting pizza.
Should I just jump in?
Yeah. Okay. Yeah, with that, we're going to invite Neha Kamat to come talk to us. She's going to be in the faculty. We're really excited to have her. This is something that we're really excited to do in GeneMods, which is basically welcome new faculty members who are in the synbio area, so we can hear about what you do, because we're all really curious. So, with that, thanks.
Well, thanks guys for the invite. It's really such an exciting club. I wish I had this when I was in grad school. So my lab is really new. We started in January, and so now we look like a functioning lab with all the equipment. But it's been really an exciting process, and it's great to be here in Northwestern too. So the question that's really motivated the work that we do is this question of how do you make biomaterials think? And over the past 2-3 decades, there's been this great emergence of these beautiful biomaterials where you have a lot of control over the physical properties. Pizza update?
How would you feel about waiting versus taking a break per week?
Yeah, end of meeting pizza.
End of meeting pizza. End of meeting pizza.
Yeah.
Okay.
Good.
You guys feel like you won't be angry?
Super hangry.
All right, okay. I'll try to be speedy then. Okay, so there's been a lot of great work with biomaterials in terms of having a lot of control over their properties and just what you can do with them. But in general, what we've designed a lot of biomaterials to do is degrade in response to different signals. And it would be great if you could make them do more, maybe make them make decisions. And so the answer to that to me really seems to be synthetic biology and the design of these great cell-free systems, genetic logic circuits, and things like that that you might be able to embed or integrate with biomaterials to create more thinking biomaterials. So that's the basic idea here. So the material of choice in my group is the bilayer membrane. And this is a really fascinating structure to me. We normally think of the bilayer membrane as this very passive structure that contains important metabolic systems. But this structure is found in every single living organism, and it really defines the boundary of our smallest known unit of life. And it is at its core kind of a sensor where it can sense different environmental signals of interest, and then it can translate that to the interior of the cell to engage metabolic systems. The bilayer membrane can also do really interesting things like undergo phase changes. So there's physical effects. It can deform, it can grow, it can divide. So it's really quite dynamic. And what's really neat about it is that it's very simple to create these systems in the laboratory. So if you take the major component of the bilayer membrane, the phospholipid, you just add it to water, it'll form this self-assembled structure that you see here, driven through hydrophobic interactions of these fatty acid tails of the phospholipid, and also primarily because water really wants to form these ordered networks, so it'll drive other things to assemble and disrupt its network. So there was a lot of interest, I would say starting in the 60s and 70s, looking at lipid-based bilayers to try to understand cells, but then they also became really popular for drug delivery systems. So most of the current carriers that we have in play for medical therapies, for both imaging agents and therapeutic drug delivery, are based on lipid carriers. And so based on that concept, there was a lot of work over the past 15 years or so, I would say, in terms of designing other molecules that would display the self-assembling quality into the bilayer. So this is a range of amphiphils that will form a bilayer vesicle. On the left, these are natural molecules that you can find in your own body. So fatty acids are actually thought to be to compose or make up prebiotic cells, our very first cells on the earth, because they're very simple. They're found in meteorites. There's prebiotically chemical routes to make them. Those are interesting. Lipids are obviously what make up our cells today. And then you have peptides, proteins, and dendrimers. These are molecules that have biological components but have had to be modified in some way to get them to assemble. And then you have dilocked polymers, which are really synthetic. And so you can choose to work with one or another of these based on what your interest is. So these molecules are very permeable. Dilocked polymers give you a lot of synthetic control. So let's say you want to make this bilayer thicker and load something really big inside the membrane. You can change the physical chain length of this block in your polymer, and you can get that. Or you can make these membranes pH responsive or light responsive by putting in a chemical moiety in here. So there's a lot of synthetic control you get with dilocked polymers, and we tend to work with these as well as fatty acids and lipids and kind of mix and match based on what we want. All right, so another reason you can work with bilayer membranes or an application is to try and reconstruct cellular behaviors. The cells display all these really dynamic and intelligent behaviors, and one approach you can take to try to understand how they do these things, like deform and talk to each other, and house metabolic reactions, is to try to recreate that from the ground up. And so the idea here is if you have fine control over what you're working with, and you can create or recreate a behavior, you really get to the heart of what are the very basic physical and chemical principles that are driving this higher order structure. And then you can also think about using those insights to build new types of technologies. And so when we go to build a cell-like structure or a biomaterial that we think, there are two components you'd like it to have. The first is some sort of scaffold. So here in our case, this is the bilayer, upon and within which you can start to build up different chemical systems. And then the second component is something that gives you computational power. So in our cells, that's RNA and DNA. And this really has the power to take different signals and translate that into the generation of completely new molecules. And so we like to think about bridging these two molecules or systems together. And this is the overall goal that we've been thinking how do you design a vesicle or a material that could respond to a specific signal of interest and then generate a secondary signal that you might use to, for example, control cellular behavior? So one of the reasons that you might want to do something like this is you might have a cell that is not responding to signals in the way you'd like, maybe an indicator of a pathology and the cell is not noticing it. Can we design this intermediate sentinel particle that can maybe go and specifically look for a signal and then generate a secondary signal that a cell will respond to? This is that kind of idea that we're trying to recreate with biomaterial. And so if you look at the system, what I realize is there's three major components that you'd like to have if you want to design a material like this. The first is, how do you control the interface? This is really the first sensory component in your system. How do you design that to respond to a signal of interest and generate a second signal that you might be interested in? The second is, how do you start to think about coupling RNA, genetic polymers, and those types of chemistry to a confined environment inside the vesicle? And then third is thinking about just how do we control some of the physical features of the system. So we like to work with microfluidic techniques and some microcontact printing techniques to really create setups where we can study these artificial cell-like materials and really study how they have the ability to engage or signal with other cells and particles. So I'll talk a little bit about that. Okay, so the first system I will talk about is an example of a responsive membrane, so designing a membrane that really responds to a stimuli of interest. And so we normally think about stimuli in terms of a chemical signal. That's just kind of how biology seems to work, but there's a lot of interesting mechanical signals. So I started thinking about how do you design a material that might tell us about mechanical forces. This could be useful from a biotechnological application standpoint. If you wanted to know when a material is going to deform or the stresses it's experiencing, it might be great if it could talk to you and tell you what sort of stresses it's feeling. In biological environments, we have a lot of different mechanical forces that could also indicate pathology. So I was thinking here about how we might first figure out if there are stresses. For example, if you had intraocular pressures in your eye, and you wanted to measure those, or you wanted to release drugs in response to stress, how do we know where those stresses are occurring? So this is an example of that. So to design a stress sensor in this membrane, what I started working with were these near-infrared porphyrin dyes. And what's unique about them is that they have this ability to twist. And so these dyes are hydrophobic. They go in the membrane of your vesicle system. And here I'm working with polymer membranes. These are dibuccal polymers that have a peg group on the outside right here, and then they have a hydrophobic polybutadiene, and they'll self-assemble into really robust, stable structures. Okay, so these dyes are pretty unique because they'll twist. And so as they twist, their photophysical properties change. And you can use that information to tell you about the environment the dye is in. So you take this dye and you put it in a very low viscosity environment where it has a lot of conformational freedom, it'll take on an entropically favored twisted state. And then if you pull it into a really confined environment, it will be planar. So you can use that information to tell you about whether a material is being stretched or not. And so this is a ratiometric method that we use, where you can determine the peak emission wavelength of the dye when it's in its twisted conformation, and you can determine the peak emission wavelength of the dye when it's planar. And so if you get a spectra from your sample, you just take this intensity ratio from the planar to twisted wavelength, and that tells you kind of a population average of the conformation. So if the dye is, if this ratio, planar to twisted ratio is above 1, it means most of the dyes are in a planar state. If it's less than 1, it means they're mostly in a twisted state. So using that, and this is just a calibration where we can put these dyes in different environments of known conformational freedom, which we use standard viscosity solutions. And you can see as you increase the viscosity of the solution, it becomes more and more planar. So we wanted to see if we could use that as a sensor for our membranes to tell us about the mechanical effects it was feeling. So this is one study where we just took our vesicle and we lysed it using surfactant Triton-X and destroyed it. We wanted to see, will a membrane reporter tell us that this vesicle is being destroyed? And so it does. It switches, the dyes switch from a planar to twisted ratio in direct correlation with the amount of Triton X that you had. And what happens is it's sort of reporting a shift in the phase or the conformation of the polymers around it. So as you lyse a membrane of any kind, it generally goes from a bilayer state to a micellular state. And so here there's a larger hydrophobic volume and the dye can twist and has more freedom. And so we can actually start to measure that with this dye. And so this could be useful if you wanted to track the degradation of a dye in the body or if you wanted to track conformational changes of the material. The other thing we found is that we could detect points of rupture or predict them in our polymer vesicle. So I had been working with a polymer system that ruptured in response to light. I wanted to use that for a drug delivery purpose. And so I started imaging it with these porphyrin dyes here. And I'm using a multispectral camera that can pick out different emission wavelengths in the field of view. And what I noticed was there was a point in this membrane that started to blue shift, indicating the dyes had more freedom there. And it might indicate pore forming. And then sure enough, we had vesicle actually rupture from that point. So this could be really a promising tool to start to detect or alert us to material failures. All right, and then the last thing that I found was that you could actually use the shifts in this dye to directly measure changes in membrane tension. So I think this is going forward going to be a powerful tool for cellular membranes, where my group is really interested in measuring stresses and tensions in cell membranes. And what you can do is you can take a vesicle or a bilayer membrane here that has this dye, and you can impose controlled tensions on it using a technique called micropipette aspiration. And as you do that, the porphyrin or the dye will shift in a really sort of predictable way in response to the applied tension. And so that might allow us a way to optically measure changes in a material without having to go in and probe it directly. So that's just an example of one type of responsive system. We're interested in the future of going into proteins and different types of ways to sense environmental cues, but I think this gives you an idea of how you can use an artificial membrane to sense a variety of different types of stimuli.
Neha,
So here are signals for, yeah.
does this dye behave similarly in phospholipid membranes, or is it polymersome specific?
So this dye is kind of neat because it's really huge. So lipid membranes are 3 to 5 nanometers in their hydrophobic pore, and this dye is 3 to 5 nanometers-ish. So we actually had to move to polymer membranes in order to accommodate this special dye, which is kind of the power of polymer membranes. But there are a variety of other molecular motors that have this behavior that I'd like to start working with to be able to see if we can get them into cell membranes. Yeah, so here this is an idea of the signal is force, and our response was actually light in this case. And so we're interested in sort of looking at embedding these materials in cells and looking at forces in a range of areas. Yeah.
I'm just curious how, if at all, does the extinction coefficient of the dye change the conformation?
It certainly does. So what mostly happens, we use the emission wavelength opposed to any sort of quantum yield or light intensity, because that can be attenuated by a lot of different things. And so we look at like lifetime of the dye or the actual emission wavelength to give us that structural information.
So, so then how is that taken into account when you're doing the ratio aspect of it? Like the difference in extinction coefficients, if, for example, the dye is absorbing more in one conformation than it is in the other.
Oh, if it's absorbing more.
Absorbing, I mean, it can also essentially end up having an effect on
so that's a great question. So there's not a huge difference in the extinction coefficient within the wavelengths range that we're looking at. It's about a 10 to 15 nanometer shift. But what we hope to do is use that calibration at the beginning and putting it in known physical environments and seeing that it shifted in a predictable way to tell us that in that case, if the dye concentration was exactly the same in those known physical environments, but the conformation, the fluorescence emission changed. So that told us that we can use that more reliably as a reliable indicator. I think that's the issue a lot of times with dyes, is that there's a lot of chemical influences over their properties, and so you end up having to use a lot of controls. But ratiometric methods sometimes can help you get around that. story I'll tell you briefly is about starting to think about how we put chemical systems inside of this vesicle. And so I was wondering how we couple RNA specifically to the membrane environment and start to replicate it, for example, inside. And this happened, so I started thinking about this question towards the middle or end of grad school, and I discovered someone who I thought was really answering this question in a very unique way, which is Jack Shostak's group up at Harvard and NGH. So they've been thinking about the idea of coupling membranes and RNA from the origins of life perspective. And how do you build a cell that may have existed on a very primitive Earth? And what's really interesting about, I think, origins of life work is what you're doing is you're essentially looking at very simple material interactions and asking how they can lead to emergent behaviors that seem much more complex than the individual components. And that's kind of what we want to do in the long run, is build these emergent behaviors from simple material systems. So the way this lab is, or the time frame, to give you a little context about where we're working, it's sort of 4.5 billion years ago the world formed, and then 3.5 billion years ago we have our first known cellular life. And so somewhere in between there, we know that chemicals started to transition to early forms of biology. And what were those processes, and what did that look like? So there's different people working in this field at different sides of this question. Some people are working on prebiotic synthesis and how you can form very basic nucleotides and things like that. We were working a little bit farther up and thinking about how a very simple protocellular system might begin to have the capability of Darwinian evolution. And that's really the ability to replicate RNA in a chemical manner and grow and divide its membrane. And so when we study RNA replication in the system, we need a prebiotically possible way to do it, which I'll mention. But the question I was asking is, how do we couple these processes, and how might you use a membrane to actually enhance a genetic process? We sort of think of these two systems as separate, but the fact that they coexist and co-evolve really leads us to believe that there's a cooperative interaction here. So I wanted to start thinking about how can we use a membrane to actually enhance RNA replication. So this is how we replicate RNA. In our bodies, we have a nucleotide triphosphate, right? That's our substrate. It goes and it base pairs with a template, and then it will link into a growing primer strand. The diphosphate is a very important group. So this whole process is driven with the help of an enzyme, the RNA polymerase. So what we want to do is replicate RNA. We were looking at how to replicate RNA without the help of complicated protein machinery that probably didn't exist yet. And so what was found in the 1960s, this is actually found a long time ago, is that you can chemically replicate RNA if you have a really good leaving group on your nucleotide monophosphate. This is nowhere good enough to replace an enzyme, but it kind of works. So what you do is you have an imidazole group, which is a really good leaving group. It's on your nucleotide monophosphate. This molecule will still base pair with a template, and then it will spontaneously link to a primer strand, and this imidazole group is displaced. So you just need some magnesium or some divalent cation, and then a primer, a template strand, and these nucleotides. And you can replicate RNA very slowly. So the question I was asking is, how can we use membranes to help or improve an RNA-based reaction? And one way we can do this, you can get some inspiration from yourselves, is receptor-ligand interactions. So membranes essentially just pull everything onto a 2D dimensional surface. Ligands will often first passively absorb onto the membrane, and now it can diffuse and find a receptor really quickly with higher probability. And so that's the idea we sort of wanted to replicate. Can we use a membrane as a surface to localize all of our reactants and then enhance their reaction efficiency? And so to do that, we have these lipophilically modified these nucleotides. And then what you can do is you can add these nucleotides to membranes. They should flip into the interior. They should pull templates that they're going to copy to the surface. And then you should have everything on this 2D surface, and it should replicate with greater speed and efficiency than a system that's in 3D bulk solution. So this is also kind of an example, back to our original goal here, of a signal response event, where now our signal is this this lipophilic nucleotide, and it's able to interact and engage with this membrane and actually initiate a form of RNA replication. This is a very simple demonstration of that idea. Okay, so this is a video where I wanted to look at whether or not these molecules actually can localize RNA templates to the membrane surface. These are phospholipid vesicles that have RNA that's fluorescently tagged in green. It's a poly-U strand, just 15 U's with a fluorescent tag inside. And I'm adding a adenosine molecule to the outside that's lipophilically modified. And so what you're going to see is this video starts when I've added this molecule, and you see the localization of these RNA strands to the membrane surface. So the green signal from the longer RNA templates over time and pretty quickly starts to move towards the membrane surface. So we can sort of change the spatial layout of these RNA molecules inside this compartment by adding the signaling molecule. That's kind of the neat power of compartments in general. So after we did some follow-up work to learn that this was actually not a base pair specific interaction, but it was electrostatic. So the imidazole groups on our nucleotides are positively charged, and so they pull RNA once they enter the vesicle to the surface through the interactions with the negative phosphate backbone. All right, and then what we looked at was, does this actually help the RNA replication efficiency? So we have these primer and template strands, and we just wanted to look at what the difference is between a lipophilic version that would pull everything to the membrane surface versus something that was rather polar. And so this is our polar nucleotide, and we can measure the reaction rate as we increase the concentration of nucleotides, and the reaction rate increases as we increase the amount of nucleotide. That makes sense. If you have this polar nucleotide, the effect of vesicles being present is very negligible because the reaction doesn't have anything to do with vesicles. It's just occurring in solution inside of the vesicle. If you have a lipophilic nucleotide in solution, you start to see this enhanced effect at low concentrations. So these lipophilic nucleotides will form, even without vesicles around, they'll form little micelles and aggregate in solution, and so they kind of concentrate, and that's what we think is happening here. If you add vesicle bilayers, things get better as well. But then everything saturates out here. And what we learned is that you're getting this saturating effect where the concentration in solution is sort of the same as the concentration on the membrane. And at a certain point, the membrane isn't going to help you increase the local concentration anymore. But it's really at these low concentrations where you can use membranes to potentially enhance chemistry. And so in an origins of life perspective, which we were interested in, we thought this would actually benefit systems that had membranes and where you have low concentrations of RNA, where you're hoping to eventually form a catalytic RNA, like a ribozyme. It would be really useful if you had a compartment to enhance the speed of that reaction and also contain the successful winner of that. Okay, so that's kind of what we're doing here. Going forward, we'd like to start expanding the types of RNA systems in here to more complex systems like riboswitches and things like that, where you could really use the sensory capabilities of RNA inside the compartment. All right, so with that, I'll just give you a few, this isn't really a story, this is just showing you some other techniques we use in our group. One of the things that we really like to do is control some of the physical features of our vesicles, especially since we're trying to confine different chemical systems inside. It's nice to be able to control the exact number of particles in vesicles, the size of those vesicles, and we can do that with microfluidic techniques. So this is a technique that was actually developed in Dave Waits' group at Harvard University, probably back in 2005 now. And what you do is you focus together 3 fluid phases. An aqueous phase is something like PBS. You have the organic phase that has your polymer or your lipid. This is actually a difficult technique to do with lipids, but it's quite nice with polymers. And then you have the interphase, which is aqueous again, and it contains whatever you want to be inside. So you can put a cell-free translation system here, or you can put, I'm putting in a certain number of polystyrene beads just to show that I can control the exact number of beads that are inside my vesicle. And then you collect these double emulsions, the solvent evaporates, and you're left with these nice millimeter vesicles of very nice size distribution. You can control the size by just simply increasing the flow rate of this outer phase, so that gives you a lot of control. And another technique we use is microcontact printing to really try to control the positioning of our vesicles. So down the line, we'd like to create systems where we can interrogate the ability of cells and vesicles to talk to each other. And so it'd be nice if you could lay them out in nice arrays and look at how the interspatial distance between different particles and cells or their size really affects those signaling dynamics. So this is an example of that. You can sort of take some biacylated vesicles, print nutraavidin in these precise patterns, roll them across your surfaces, and your vesicles can get isolated onto these islands. And you can control things like the size and positioning of your vesicles. So I kind of showed you 3 stories for thinking about designing these thinking biomaterials. And there's a few ways, a few advantages of vesicle systems. So you can really design the membrane to be responsive to a variety of different stimuli that you're interested in, even using non-natural components like orphan molecules to really help you build these sensors to the best of your ability. Thinking about engaging different chemical systems inside and using microfluidic and microcontact printing techniques to control their physical properties. And so right now what my group has been working on is thinking about designing these systems that are continuously acting. So how do you design a material that doesn't just fall apart, but rather can sense a signal, engage a chemical system, and respond, and do this over multiple periods of time. And we're thinking about designing this bilayer. There's a lot of, right now we are working on trying to embed different membrane proteins in this to really expand the type of signals you could be interested in, and also how to translate those signals to the interior. We're interested in synthesizing biological materials inside these vesicles and thinking about how compartmentalization can be used to enhance those reactions. And then we have a whole other area of just stress sensors. So that's totally different. But I think that's it. If you guys have any questions.
How does the size of the vesicles affect the properties you're studying? Because like, it looks like most of your vesicles are like 10 to 50 microns, and bacteria are like 2 microns.
Yeah. So so the 10 to 15 are really useful when you want to do microscopy. But what we actually do is you generally will do build large vesicles for imaging purposes, and then you can make the vesicles on the nanoscale and sell, bacterial-like sizes to do bulk studies. So there's kind of this tool side that you do. But I guess cells are actually, mammalian cells often are on the micron scale, a little bit larger. So it kind of varies with what we're trying to model. But we kind of work on both because drug delivery purposes, you want nanoscale. You can't put a 10 micrometer vesicle. Yes?
The second story, what kinds of molecules can you flip into the vesicles? Is it, can you compartmentalize just about anything that you attach to the right type of polymer?
Yeah, so you, the only things that can flip across membranes in that way, in that sort of particular way, are something with a single fatty acid tail, and your head group can't be too bulky because there's an energetic barrier to pulling it across. So for example, phospholipids with 2 diacyl chains will not flip very slowly across the membrane, even in a cell. It happens on a seconds to like days time scale. The fatty acids have this like really beautiful and fast signaling property. And then I think you'd be limited by the chain number and that bulky head group. So this actually this was fast, but I think you couldn't go too much larger than a nucleotide. But I think there's other ways to get things in. So there's, you can use so there's been some interesting demonstrations so far with theophylline that actually can, so another group was trying to show that you can engage a riboswitch inside a vesicle. So they used a theophylline riboswitch, and theophylline is membrane permeable. So you use membrane permeable molecules. But then that doesn't give you a lot of specificity, so I think membrane proteins are probably the best if you want to be specifically sensing a specific chemical. Yes?
So I had a question. So you mentioned that the RNA is sort of drawn to the membrane because of the positive charge of the imidazole group.
Yes.
But it looked like your construct was actually had like positive imidazole, but then right next to the negative phosphate, like a Zwitter ion. So
Yeah.
is it set up like a giant dipole or something?
So what we did, I actually have a ton of supplemental graphs because we thought it was going to be base pair specific, and I was actually following up on a former student who said it was for sure base pair specific. So we had to do extra work then. So it does seem. So what we did was we used zeta potential measurements to look at what that surface charge looked like, and we thought it seems like the surface charge is actually negative when you have that phosphate on there. But for some reason, it's I think closer to the membrane, that positive charge is able to still pull the negative anions there, and that phosphate group is not a deterrent. What happens also over time is that these molecules hydrolyze, and so the phosphate and nucleotide diffuse away. Where is that? Yeah, so this group will hydrolyze over time. So you just sort of build up positive charge. But yeah, I think it's intriguing.
So a couple brief announcements before I let you get your pizza. First, tomorrow, our congresswoman, I believe, is coming to talk about science advocacy. So be sure to keep an eye out for those. You'll probably get bombarded from all the listeners if you're interested in science communication and science policy. Second, next meeting in June will be a recap of SEED, the conference. Who all is going to SEED? Okay. So we're going to ask everyone who goes to prepare a brief 2 or 3 minute summary of something you found really interesting and present it to the group. And we'll kind of have a nice recap of all the cool stuff at SEED. And I feel like there was something else.
Pizza.
Okay, pizza.
Pizza time!